Okay, and we're live again. Welcome back, everyone, for the first week's last Q&A. You made it through the week. That's super exciting. And it's my great pleasure here to have our amazing panelists. First of all, we have Sara Solla, who is a professor in physiology and also in physics and astronomy at Northwestern University. We have Byron Yu, who is a professor in electrical and computer engineering and also biomedical engineering at Carnegie Mellon. And finally, we have our very own Carsen Stringer, who is a group leader in the Howard U Medical Institute at Geneva Research Campus. So I would like to, so welcome, first of all. And then I would like to maybe give you a few minutes to briefly introduce yourselves, and maybe each of one can say something. We've sort of done this like every day. Maybe each of you can say something about what is special about today's topic for you, for your research, or maybe you have an anecdote or where a funny story to tell us about dimensionality reduction. So maybe we're going to go in the reverse order now. Carsen, would you like to start?
Sure. I don't think I have anything funny to say. But I'm Carsen. I'm a group leader at Geneva, and I work on the visual system. So looking at how the visual system processes complex images. And that kind of work really requires dimensionality reduction because we're taking large recordings of thousands of neurons, and we need to understand how they're representing high dimensional data as well, which are images that are coming into the brain. And it's fun because we work in mice. So it's fun to think about the kinds of images that mice care about and what kinds of images we want to use to show the mice and to use for our dimensionality reduction and so on.
Great, thank you so much. Byron.
Great, I'm Byron Yu, and let's see. So we've been using dimensionality reduction in various studies in different parts of the brain. I think sort of looking back the last 10 years or so, people started off by using dimensionality reduction mostly for visualizing their data, which is one good use of dimensionality reduction. And then more recently, people started to develop hypotheses that requires some sort of dimensionality reduction or some sort of population activity to test, which is great as well. And I think really exciting direction for future research is to think about designing experiments that really leverage dimensionality reduction and maybe even integrating dimensionality reduction in the experiment, the rig code or the way in which real-time processing is going on during an experiment to gain insight that would be difficult to gain otherwise.
Great, thank you. And Sara.
Hi, I'm Sara Solla. I work at Northwestern University, as Gunnar said earlier. I am a theoretical physicist by training and a theoretical and computational neuroscientist by choice, by vocation. All these questions of dimensionality reduction to me arise from first of all, from a long-standing collaboration with Lee Miller, my colleague at Northwestern, who does these multi-electrode array recordings that Byron talked about on monkeys, on awake and behaving monkeys. And through him to have been introduced very early on to population data where we record from many neurons simultaneously. I mean, not many compared to the number of neurons that participate on the task, but many compared to 20 years ago, when we could only record one neuron at a time. And I believe that the concept of population, as opposed to looking at single neurons, is really what we need to look into if we want to understand how the brain computes, if we want to understand the brain as an organ designed for processing information. So the neurons are collectively, and all the behavior that we see, the actions, our own perception of our internal states, it doesn't arise from single neurons. It arises from the coordinated activity of a very large number of neurons. And so there was a nice paper by John Cunningham fairly recently about moving from a neuron doctrine to a population doctrine. And I'm certainly on the population doctrine end of things. And then within that context, I think of dimensionality reduction as a tool for unveiling the true modes of the brain. I believe that neurons don't act independently because they are interconnected and they act in preferred patterns that are joint patterns of activity. So I don't think of dimensionality reduction as a way of denoising or as a way of creating nice 2 or 3 dimensional images that we can observe. But I believe that these neural modes really capture the intrinsic dynamics of the brain, and they are a manifestation of the connectivity of the network that creates these preferred patterns of collective activity. So I believe that they are conceptually a building block of the population activity that we see, and therefore far more fundamental than to say it's for denoising, it's for visualization.
Great, thank you so much. Okay, I think we're ready for some questions. Students have had lots of time and put in quite a few questions, so let's get started. The first question is by Igor Zverev. How to choose a nonlinear dimensionality reduction technique for your data? There is like 29 different algorithms mentioned in Wikipedia. That's quite a lot. What are your general recommendations related to choosing an appropriate algorithm out of this diversity? Thank you. Who would like to give that a go?
I could get started if that's okay with you, Gunnar. So it is very true. You know, when you have principal component analysis, it's principal component analysis. Everybody agrees what it means. Everybody agrees how it should be done. And when we say I'm doing principal component analysis, there is no ambiguity. When we say I would like to do nonlinear dimensionality reduction, it's like you open a big Pandora box and there are so many methods, and how do you choose which one? One of the considerations was referred to today in one of your tutorials, where it was mentioned that with methods like T-SNE, or also in methods like isomap or local linear embedding, you cannot construct a reconstruction error because you cannot create the inverse map. You cannot go from the coordinates on the manifold to the coordinates in the original space. So if you want to overcome that difficulty, then autoencoders are a great tool, because autoencoders are networks that allow you to go from the high dimension to the low dimension, and then again from the low dimensional to the high dimensional. So the high dimensional, the output gives you the reconstructed vector x hat, and the input is a vector x, and then you can compute reconstruction errors. So if that's important to you, then these autoencoder networks are a very good tool. For me, I like isomap a lot within this limitation of not being able to reconstruct. And the reason that I like isomap is because there is a continuous way of going from isomap to PCA. So as I increase the size of the neighborhood in which I'm making a linear approximation and I compute distances as Euclidean, if I take that to be the whole data set, then I'm back at the PCA limit. And as I decrease it, I get back to the nonlinear geodesics that are characteristic of isomap. So I like isomap because it allows me to anchor the calculation in one end. It gives me this kind of sanity check, in which at one end of extreme end of isomap, I know what I'm doing. I'm doing PCA. So that's a bit my personal toolbox is autoencoders and isomap.
And since you mentioned autoencoders, that is actually being covered on the very last day of Neuromatch Academy.
Great.
There you go. Does anyone else? Would anyone else like to contribute anything to that answer?
I can add something about T-SNE. I find that T-SNE and UMAP, for instance, work a bit better in my experience with neural data in terms of capturing kind of the local manifold of correlations. So I tend to use them more for visualization than isomap.
Great. Byron, what's your experience?
Carsen and Sara have a lot more experience with these methods than I do. One thing that I mentioned in the intro talk was one should understand these methods before using them, and don't just like blindly like take some nonlinear dimensionality reduction method and expect it to do some magic on your data. Yeah, different methods make different assumptions and have different requirements. And one should think carefully about whether the data that you have at hand are satisfy the requirements. And if not, then one should think carefully about what I mean, one should always think carefully about what the outputs mean, but one should be especially cautious, right? If the requirements are not met, right? So I would say use with care, but when used properly, they can be quite powerful.
And I guess that is true for any analysis technique really you want to use, or any modeling technique you want to use. You always have to know first whether it's actually applicable, and whether and if it is, what potentially you can get out of it and what the limitations are.
Actually, even for PCA, that's a very valid thing to remember, because essentially PCA is modeling your data as a multivariate Gaussian. So if your data has a bump here and another bump there, PCA is going to do something very weird, like giving a mean in a point where there is no data. And so there is always assumptions built in the model, and you should always be aware of them before you actually go ahead and use the model.
Yeah, and that is essentially another one of those rules. Always look at the raw data. Don't just look at summary statistics, because you will miss like multiple bumps, for example, and things like that. Great, thank you so much. Okay, next question is by Siwi Bai. And Siwi asks, can I combine GLM with dimensionality reduction? If a set of variables, say fMRI data, gone through PCA, component selection and reconstruction, can we interpret the still prominent variables as features or featured variables and use only these variables for regression, for example, with behavior?
Yeah, so there have been a number of studies which have done that, right? And so one of the motivations is, let's say that you want to focus your analysis on dimensions of activity that have the greatest variance, right? So in that case, what you can do is to first apply PCA to your data, right? And then take the dimensions that are identified, and then continue with your next level of analysis based on those dimensions identified. And so that can help to avoid having regression depend on activity dimensions with low variance.
But that can also quickly result into a fishing expedition, right? Where you essentially try that with every single principal component, and then do you want to comment on that?
Yeah, so I think the nice thing about PCA is that the dimensions are ordered based on the amount of variance explained.
Yeah.
So you can then say, okay, is it the case that the dimensions that explain greater variance are maybe more helpful in explaining behavioral variables?
Right.
And this opens a very interesting question about PCA, which is, we always think larger variance is what needs to be kept, and smaller variance is what doesn't need to be kept, because we think larger variance is signal, smaller variance is noise. But when you begin to correlate with external variables, you might find surprises along those ends, that some dimensions, some principal components that you would have tended to discard because they have smaller variances associated with them, are actually important in establishing a functional relationship to a behavioral variable, or to a muscle activity, or to something which is outside of the neural system that you're looking into.
Carsen, anything you want to add?
No, I mean, I use PCA before doing regression in a similar way to Byron, in terms of in part to reduce the dimensionality of the data to regularize my regression, and also then you have a tool by which you can see how much variance is explained depending on which PC you use.
So how do you, because I'm sure that this is one of the questions that came up earlier, but some of the students here might also have that question. How do you, when you do that, how do you choose the cutoff?
Yeah, in my case, often I'll use a cross-validation technique to choose the cutoff. So you can take the principal components from one half of your data and project them onto the other half of your data and see how much variance they have. And so that's a way for you to be able to see how much of that PC actually is noise variance in a sense, and how much is repeatable throughout your experiment. Yeah, so that's one way to try to tell if it's really a noise PC in the sense that it's not repeatable. It might be independent noise among neurons, rather than a PC which contains correlated activity across neurons that's repeatable.
Great, thank you. Okay, next question is by Sarah Morsi. And Sarah asked, how to determine the best perplexity number in the T-SNE algorithm? This is a very popular question.
I can answer that if you want.
Sure.
There's no right answer to this question, but there's a few different benchmarks you can use for T-SNE. For instance, you can, you're not going to use variance explain because you're not doing a reconstruction of your data, but you can try to see that in this low dimensional manifold that you've created, if the points that you've embedded are in have neighbors which are similar to the neighbors in your original space. So say two neurons are correlated in your original space, then hopefully the T-SNE puts them close to each other. And if it does a poor job, for instance, of doing that, then it might not be the best perplexity that you chose. And there's other metrics like that in terms of preserving local and global distances that you can use to try to decide what the best perplexity is for you to be able to visualize your data.
Does anyone want to add something to that? I think the original paper, the author of the original paper essentially says, use anything between 2 and 50. Quite literally, which is sort of like, well, give me a ballpark, but it's not very precise.
A good kind of like benchmark too, which is recommended in a recent paper by Kobach and the Barron's lab, is that it should scale with the number of data points you have, because perplexity is approximately proportional to the number of neighbors you're kind of considering when you make this T-SNE graph. So you probably want to use bigger perplexities when your data set is larger. But you still want to play around a bit with it for sure.
And you've already kind of mentioned this, but this is also a question that came up earlier that was related to the T-SNE algorithm. What does it actually mean? What does it do? What's the intuition behind it?
Do you want me to keep, someone else can answer?
Yes,
no,
please. I think you have most experience with T-SNE of the three of us.
I think I don't know how far back we want to go, but the idea is that you're not doing a matrix decomposition, you're not doing a matrix reconstruction algorithm. You're trying to reconstruct your distance matrix in your original space, which you can think of as your correlation matrix between neurons. So you want to put correlated neurons in your original space, like maybe you have a 10,000 dimensional by 10,000 dimensional neural matrix, and you're trying to compress that into this 2-dimensional space. So neurons which are correlated here, you're going to try to put close to each other in the T-SNE. And you're in this, by doing this dimensionality reduction, you're always losing information because you can't embed 10,000 dimensions in 2. But because it is nonlinear, you can kind of mush stuff around and move it to try to put things close to each other that are correlated in more so than you can with a linear reduction method. So that's where the nonlinearity comes in, that you can kind of morph the space a little bit in the T-SNE. I don't know
to add anything to that? It's one of those magic tools. Themes.
So, Carsen, since you have used it much more than I have, how much does it bother you, the stochasticity of the algorithm? The fact that if you run it again, every time you get something different, as opposed to isomap or one of those techniques?
So actually, if you run it with the same initial conditions, it's not.
Well,
yeah. So it's a question of like, with every algorithm which doesn't have, which has many local minima that it could find, you want to try to find the best initialization. So it is a good question. And often it's recommended to use PCA as an initialization because that's going to start you off in a good place for your global structure. And then when you run the algorithm, hopefully the local minimum that you find is closer to that global structure. But yeah, it is a problem too.
I guess the same reservation applies to an autoencoder, right?
Definitely.
You'll find different trained networks depending on your initial assignment of values for the weights. So, yeah.
Great. Byron, any other comments about this?
No.
Good. Then we can continue to the next question. Okay. Sunanda Srikant asks, why do we perform denoising by projecting the noise-corrupted data onto the basis vector found from the original data set instead of projecting onto basis vectors obtained from the noisy data? Who would like to give that a shot? So this is particular to the tutorials that they did today.
most of the time we don't have, we only have the basis vectors obtained from the noisy data. I mean, in a real life situation, right? We don't have a knowledge of what directions are the noise-free directions. If we do, of course, we can put it in by hand. So again, we find the principal components, we rank them according to the corresponding eigenvalues, and we make the assumption that the large variance directions correspond to signal and the low variance directions correspond to noise, and those are the ones we truncate. So, but the large variance directions that we project onto come from looking at the data set, and the original data set was signal plus noise, and signal and noise, I should say. It's not necessarily additive noise, but it's corrupted data by noise.
Would anyone like to add something to that? So I think one of the main reasons in the tutorial was just for simplicity.
Yeah.
If if in a certain way it's an estimation problem, if you just assume that your basis vectors are well estimated, and very reliable, then you can do the correct projection here. But really, this was just part of the setup of the tutorial to make it a little bit more approachable. As Sara said, like in practice, you would of course never have, or very rarely have, ground truth. And therefore, you would just have to deal with the noisy data.
Right.
Yeah, great, thank you. Okay, Miguel asked the next question. Calcium imaging analysis tools I've seen in rodent often will perform a PCA followed by an ICA, or non-negative matrix factorization, to identify cells. What's the logic here? Why do a PCA followed by a nonlinear dimensionality reduction? In brackets, in this particular case, to find cells glowing over time with the camera. Okay.
Do you want
Carsen,
me to answer that?
Like
yeah. So that's partly to make the problem more tractable. Is you do this PCA first, and then the problem doing something like ICA or non-negative matrix factorization, which this optimization is much harder to do. You're going to want to use a smaller set of data. In practice, in Sweet2P now, we actually don't do PCA anymore. Sweet2P, I'm sorry, is a calcium imaging processing pipeline that I've co-developed with Marius Pakitariu. And currently, instead of using PCA, we actually bin the data in time, and we instead look for we look for these kinds of sparse events that are going on, rather than reducing dimensionality, which might kind of miss some of these things. But it's kind of, there's multiple ways to do this problem, and all of these solutions are useful for different kinds of data.
Who would like to add something to that?
I think that's exactly right. I mean, it's a cleanup process first, right? And that allows you to go to a lower dimensionality where these more numerically involved algorithms have a better chance of working than in the original, particularly if your original data set was very high dimension. It's necessary, really, to go to something more manageable. Manageable not in terms of number of points, but in terms of the dimensionality of the points.
Great. Byron, any comments?
No.
Sorry, I keep prompting you.
Yeah.
So
doing a great job at answering this question.
No, I agree. Since ICA came up, and earlier there was a question about dominant dimensions or dominant principal components, maybe we can have a quick comment about the use of ICA for example, artifact removal, where it's sort of the opposite way around. Like the artifact that you remove is very often the dominant one. And then, for example, in EEG, right? Like as soon as you blink your eyes, it's like the whole cap essentially just goes like whoops in the signal. Does anyone want to comment on that?
Maybe Gunnar.
Maybe I just said something. Yeah.
Yeah.
So sometimes these techniques can be used to remove physiological noise, for example. Typically, ICA is used for that, for example, and in that case, the physiological noise components, typically in amplitude, are much larger than the signal that you actually want to analyze, which is the small brain signals, for example, in EEG, right? And that comes, of course, with a whole bunch of caveats that you want to make sure that you do not remove any principal components or any independent components in that case that are actually meaningful signal, and there's no real way to control for that. So it's always a sort of like there's a flip side to that. You can clean up the data, but you might actually lose some things. And then ICA is based on the fact that you have essentially the same signal at a different amplitude across all the recording sites, for example, right? But if that is something meaningful electrophysiologically, you will also remove that.
All right. I think one important thing. Oh, can you hear me? All right.
Yep.
So I think one important thing that your comment points to, Gunnar, is that not every dimensionality reduction method has its own objective, right? And has its own definition of what it considers to be signal and what it considers to be noise. Right? And so when faced with any particular problem, one should think carefully about all the different dimensionality reduction tools that are available and think which one has the definition of signal and noise that best matches with the needs of the current problem that one is faced with. And so if one chooses the wrong method, then what one risks is that there's an incorrect separation of what's considered signal and what's considered noise, like what Gunnar was just referring to.
So in some sense, PCA is producing an output that will be the input to the next algorithm. So you have to keep in mind what are the goals of the next algorithm or the next analysis that you're going to use. So in some sense, it's like bringing a little bit of a supervised guidance to how you're doing the PCA as opposed to doing it in a purely unsupervised manner.
Exactly. So in that specific example, then if high variance equals signal, right, or high variance equals stuff you want to keep, right, then doing that is appropriate. But one could imagine in many scenarios where that is not the best thing one could do.
Yeah. Perfect. Okay. The next question is by Michael. Are the nonlinear dimensionality reduction methods mainly used for visualization? Or are there instances where, for example, you would want to conduct downstream analyses to a T-SNE result?
Well, I don't know about the T-SNE result, but certainly we have done downstream analysis to isomap results. And for instance, there's this beautiful work on the head position coming from Ila Fitter's lab recently, where she finds a one-dimensional, extremely nonlinear attractor in the form of a ring that allows her to interpret the head position data. So, of course, having one-dimensional manifold is very useful for representation, but that's not, again, I don't think that visualization is the goal. I think that the goal is to really extract few meaningful underlying dimensions that are the generative model for the data, really. I'm going to be provocative and paraphrase Surya Ganguly here, who said once to me that single neurons are nothing but a random one-dimensional projection of the population activity. So what matters is really the population activity, and you want to project it into directions that are meaningful in the sense that these are neurons that co-activate, either positively or negative, but that have correlated activity because of the connectivity of the network. And we use these reduced representations as inputs to the brain-computer interfaces that Byron was talking about all the time. We don't use as input the full neural data. We do a dimensionality reduction, we find the manifold, and then we define the VCI interface using the manifold data as inputs, and it works very well, and it's cleaner, it allows us to see better what's going on. So yes, well beyond visualization, in my opinion.
Carsen, Byron, anything you want to add to that?
I will say that you have to be careful when you interpret these, like we were saying before. Like, I think Byron has a lot of nice work on the causality of these things in terms of, is this actually a manifold that the neural activity is sitting on that it is actually exploring actively, or is it an artifact of the way that we produce the data?
Absolutely.
Yeah. And I mean, if particularly talking about T-SNE, of course, we talked about the stochasticity of the algorithm earlier. That is, of course, an issue if you take that into an analysis pipeline where you sort of blindly follow that by something else.
Yeah, I want to say. Can I say something quickly?
Yeah.
Just about T-SNE. I really like to use it in a way that not everyone does, which is where you use it to sort your raster of neural activity. And you could do this with any nonlinear embedding algorithm. And then you get these nice pictures of your data, and then you can go in and kind of look at groups of neurons or clusters and so on, and try to localize them that way. And so that's a type of visualization that allows you to look at the raw data while doing a dimensionality reduction.
Great. Next question is by Alan Zhang. And it's a very open-ended question. Besides single neuron activity, can we use dimensionality reduction to discover intrinsic manifolds in other modalities, such as fMRI, EEG, ECOG, etc., using similar techniques introduced in the outro? Byron, you want to start with that?
Yeah, I think absolutely. What one would recover in that case, or what one would be studying in that case, is sort of these low-dimensional spaces mean sort of something different when we're looking at the whole brain scale, for example, right? So whereas when looking at single neuron recordings, these low-dimensional axes represent the way in which neurons covary with each other, like Sara and Carsen were saying. If we're looking at, say, EEG or fMRI activity, maybe that would be studying more like how different brain areas are covariant with each other, which is also very interesting. So yeah, absolutely.
We use them, for instance, to analyze collective recordings from muscle activity, again, to try to find particular sets of muscles that seem to be co-activated together. Although that can be very task dependent, of course, but again, it allows you to find these modes, and some people have used some of these methods to analyze behavior. There is the famous case of the worm, the eigenworm, the eigenmodes for the worm, and people have used it also. I think it's Josh Shavitz at Princeton, if I remember correctly, to analyze the characteristic fly patterns of a fly. So yes, any kind of activity that indicates a collective, many degrees of freedom, but that you think that a few degrees of freedom are coordinated in a given way to produce the behavior that you see is a good territory for using any of these dimensionality reduction techniques. Always start with linear, see how far can you get, and then go non-linear once you understand what are the limitations of your linear method. That can also provide some guidance on which non-linear method to use.
You can really apply that on any sort of high-dimensional signal that you have. Yeah. So people have done it, for example, for muscle activity. If you were to record, from 100 muscles, for example, and you will see that there's things that people call synergies, for example, where different muscle groups tend to work together all the time. You can do it in behavior. If you, for example, record all the different bone or finger movements, right? Yeah. And then you do that during a few tasks, you figure out that you do not actually use all the degrees of freedom that you have, all the combinations that you theoretically have, but only a subset of those. So again, dimensionality reduction can be used to look into that. So it's really like a very open and powerful technique in a certain way. As long as you have multidimensional or high-dimensional data, there's usually a way or a dimensionality reduction technique that would make sense to look into. Okay. Let's see, what do we have? So here's a question by Nida Kundryashova. Sorry, that just moved. So Nina asks, what are the open questions in the dimensionality reduction field? Are there any examples of the data sets or manifolds where all of those methods mentioned in the intro lecture fail to detect the intrinsic structure? And I guess we have to assume that there is intrinsic structure that's discoverable. Who would like to give that a shot?
I think this is a really fun question, or a really thought-provoking question, because we don't know the answer to it. And yeah, so, because we don't know the ground truth, right? We could be either a little bit off track or entirely off track, right? And what's important in the studies that we do using dimensionality reduction is to try to ground what comes out of a dimensionality reduction method in terms of something tangible that, for example, ability to predict behavior, or some association with something you can measure in the outside world that the dimensionality reduction method didn't know about, right? And if it can recover that, then that's maybe some indication that what's being identified by the dimensionality reduction is identifying something meaningful or reasonable. But beyond that, I think this is a really exciting direction, for the field, which is to try to figure out to answer that question.
And the main unsolved problem is noise, really. It's how these methods fail when the data is very noisy. Somebody in the chat section was mentioning tokens, embedding, and other ways of doing dimensionality reduction in the dynamical system, going from T to T+1, as Byron was mentioning, in the construction of the brain-computer interface. All this is very nice as long as your data is relatively clean. Once the noise distortion becomes important, all these methods tend to take you into the wrong path, which you know by testing them into data where you know what the ground truth is. But when you are faced with real data and you don't know what the ground truth is, sometimes you don't even know that your method is failing.
Yeah, and I think a really interesting thing about Sarah's your comment is that we don't have a good definition of what's noise, right? So what's noise to us today may not be noise to us, a year or two from now, when we can explain that variability in that.
Exactly. Noise is a fluctuation whose origin we don't know, and whether it's really stochastic or due to a hidden variable that you're not monitoring or aware of.
Right.
Yeah.
Right.
I was going to ask you about your definition of noise, but you just gave it to us. That's great. Does anyone have a different definition of noise? Just out of curiosity, because people tend to have very different definitions.
I think in my work, I tend to think of noise as anything that's not correlated across the population, which is similar to so it might not be, there's definitely correlated noise that's related to other variables that you're not looking at. But if there's independent, like Poisson noise, for instance, that the neurons are having, then that's the kind of noise that I would consider throwing out.
Right. Yeah, I usually think like my working definition would be, the same as yours, Carsen. And the thing that's always in the back of my mind is, if we could record for more neurons, would the variability of some of the neurons I'm looking at now, would they be explained and no longer be noticed to me?
Right.
Yeah, it's a great question.
This is always the question mark, that's.
Great. Thank you so much. Okay, Miguel asks, could you go a little deeper into linear discriminant analysis, or the nonlinear method we didn't quite get into in general? Not quite sure what the latter refers to, but I guess linear discriminant analysis is specific enough. Now, I'm not sure how much deeper Miguel would like us to go here, but who would like to give maybe a sort of intuitive explanation of linear discriminant analysis?
Well, that's the easy part of the question.
It is, yes. Okay, you're welcome to give it a deep dive too, Sarah.
So what linear discriminant analysis tries to do is, again, to find a direction in this neural space, so that along this direction you have maximal variability of an external quantity that you are monitoring. And if it's, for instance, going a binary classification going from left to right, then you expect that as you move along that axis, you project all the data on that axis, there will be a threshold so that everything below the threshold is left and everything above the threshold is right. But you could also have a gradual variation represented by a sigmoidal function or an exponential function or your favorite nonlinearity or even a linear dependence, which tells you how an external quantity varies continuously as you move along that line. So the idea is that there is a line that you project all the data onto that line and is maximally informative about predicting this external variable. It could be binary or continuous. At least that's the way I think about it. Maybe maybe Byron or Carsen think about it in a different way.
Do you? No? Awesome. Thank you so much. Next question is by Ariel Yu. Ariel asks, how do you denoise if you do not have eigenvalues vectors from a clean data set? I believe the example we had for our tutorial had us project the noisy data set onto the eigenvector's original clean data set. But what do we do if we do not have a clean data set?
Yeah, so it depends on what types of eigenvectors you're looking for. If, for instance, you're showing many different visual stimuli many times and you have many trials, you can take trial average data, and that can reduce the noise in terms of the PCs that you try to get. If you're looking at ongoing activity, you don't have that luxury, but you can try to use as many time points as possible, for instance, to estimate your eigenvectors, and that can help to reduce your noise. Yeah. Always having more data is always better.
Surprise.
Yeah. That point was really nicely made in the tutorial, that PCA works better once you do trial average first, and then you work on that data. Presumably that data is cleaner and allows you to find these relevant directions, which are the ground truth relevant directions, because part of the noise has been averaged out.
Great. Okay, next question is by Yvonne Lee. How do you recover a S-shaped manifold? We know PCA doesn't work, and T-SNE doesn't seem to be relevant either.
You do isomap. I mean, isomap is famous for doing this how do you call it, this bread roll or this roll-shaped thing, and it works with an S also. I mean, this is really what it's designed for. It's a great success of isomap, is to be able to deal with such surfaces, where the main thing is a curvature, and it's a curvature that varies smoothly as you move along the manifold.
Anyone want to add something to that?
In theory, like other dimensionality reduction methods will work the same depending on how many neighbors you define.
That's true.
But then again, it will depend very much on the perplexity. But yeah, sorry.
Right. And if you define, for instance, in the case of the roll, if you define anisotropic neighborhoods also, so not just an epsilon ball, but an ellipse, that allows for more neighbors in some direction than in other directions. But then those things just become so case specific that it's very hard to give a general prescription for how to do. It's I think nonlinear dimensionality reduction is more of an art than a science. The more you do it, you begin to develop an intuition. For this problem, this method is likely to work because you've done something similar before, right? So, but it's not based on theorems, really. It's much more heuristic than that.
Awesome. Oh, long question is next by Alex Ivanov. Alex asked, I really liked the BCI example from the outro video showing us how we can simplify the system in order to study it. Byron, you used linear relationship between the neural activity and the cursor that was controlled. How do we know that? First, scaling down the number of output neurons, and second, getting rid of the nonlinearity, so these two aspects, is not changing qualitatively the way the system works. Can we be confident that our findings will apply to the real brain context?
Yeah, this is a great question. By defining this linear mapping between neural activity and cursor movements is, we acknowledge that to be not trying to mimic the actual relationship between the activity of these neurons and arm movements, for example. And that relationship is, if we wanted to predict, say, the details, right, of the arm moving with nonlinear muscle dynamics and joints, right, that's a nonlinear relationship between neural activity and arm movements. So sort of part of the design here of the system is that we specify a linear mapping, although we could also consider specifying nonlinear mappings between neural activity and cursor movements. I think it might make it a little more difficult to analyze, but I think there are some interesting experiments that could be designed in that vein as well. Okay, so I think that answered one part of the question, maybe. And then the other part was about reducing the number of neurons or something. Is that right, Gunnar?
Scaling down the number of output neurons, yeah.
Okay, I'm not entirely sure what that means. Does that mean maybe doing dimensionality reduction on the neural activity and then using the low dimensional factors to then estimate cursor movements? And so maybe the question was, why did we do the first dimensionality reduction step? Maybe. Okay, so in the design of that experiment, we did that to dissociate between these two types of mapping switches in the Sadler et al. Experiment. So without having first done factor analysis and then doing the Kalman filter, we would not have been able to define these within manifold perturbations that are different from these outside manifold perturbations. If you're wondering about how the subject's ability to control the cursor depends on how many factors are used, we systematically varied the number of factors that we used in the experiment. And what we found was when we used a large enough number of factors, the decoding performance was very similar. So we started at a large number of factors, and then we went smaller and smaller and smaller, down to 10. And then when we got down to 6 factors, that's when the cursor control performance started to degrade. Okay. So I think what that's saying is that there's structure in the neural activity that you need to capture. And if you don't use enough factors to capture that structure, your decoding performance is not going to be as good. But if you use more than a number of factors, at least in our experiment, we didn't see any degradation in performance if we use too many factors. If that makes sense.
Great. Thank you so much for clarifying that. We have time for maybe one more question, and then we should wrap it up because we're coming up to the hour. So here's another question by Yvonne Lee. How do you know if your data is nonlinear? Plotting doesn't seem feasible when there are hundreds and thousands of dimensions. Who would like to give that a go? That's a very popular question. We had that earlier today as well.
Yeah. So it was it you, Byron, who showed this way of generating many different two-dimensional projections in your tutorial?
Uh-huh.
So I think that's a good tool. I actually start by doing a lot of one-dimensional projections, for instance, to see if the data is unimodal or bimodal. We do a lot of those plots. Now, of course, if you have 1,000 dimensional variable, that means 1,000 one-dimensional plots and of the order of 1 billion two-dimensional plots. So it's not a very practical, but that's what you start doing to visualize the data. You can use in the one-dimensional projections to get some guidance about which two-dimensional projections to look into, and maybe that gives you some guidance of what three-dimensional projections to look into, because you have this tool for rotating the two-dimensional projections into one another. But yeah, that's a problem. That's the problem of high-dimensional data.
Anyone want to add something to that? Any tricks? How do we know data is nonlinear?
You can. It depends on the data, definitely. But if your scree plot decays relatively slowly, for instance, or like there's a lot of significant weights at the later PCs, then it's likely that maybe there's some nonlinear manifold underlying it. But that might not be the case. Could be like a very high dimensional linear space. But if you have a lot of significant components, it could be that it's this nonlinear manifold that's taking up all of these planar dimensions, but is really something lower dimensional that you need to figure out. And to really kind of explore data like this, I think it really helps to think about the external correlates. So thinking about what images were you showing, for instance, the animal, what behaviors was the animal doing, and you can use these to subset your data. For instance, which parts of your data are predicting which external correlates, and try to use that to start to explore your data and lower dimensions potentially.
And another possibility is to try to look a little bit at residuals. So imagine that I project all my data in a two-dimensional plane, right? And if the data is fluctuating around that two-dimensional plane, then if I take an arbitrary direction and I look at residuals as a function of that moving along that direction, I should see fluctuations that look random. But if the data has a little bit of curvature, it was a little bit like this, and I tried to flatten it into two dimensions. Now, no matter along which direction I plot the residuals, I will see residuals that are positive, go to zero, and then I get positive again. So, that could give you a little bit of a handle. So if you start linear and then you begin to see what you're missing, it might give you an intuition on in which way you need to go nonlinear. But again,
Great.
as I said before, I don't think that this is a good, unique mathematical prescription for how to do that. And we all develop intuitions on how, based on experience, on how to do that for specific types of data.
Yeah. Great, thank you so much. I think we should end the questions here. For those questions that we have not answered, please copy them over into the NeuroStars forum. That way you will get an answer there. And maybe before we wrap up, maybe I can ask each of our panelists to say a few last words of advice or wisdom related to dimensionality reduction or other aspects of research and life. Maybe, Sara, do you want to start with that?
Sure. So I think I would emphasize the things that we talked about. Always plot the data, get an intuition of what the data looks like. Always start linear in your analysis and see how far you can push it. And hopefully that will tell you in which way to go non-linear. And about as for life, always enjoy your work. That's what keeps them working. Enjoy your work even when things are not coming out the way you expected them to come out. And I really like Byron's tutorial example today when he said, okay, how does the data migrate from this BCI direction to this BCI direction? And my intuition would also have been that it would migrate, that way as opposed to reorganizing itself the way it did. So sometimes the failure of your hypothesis is a very good opening into a better way of thinking about the problem. So even when you're wrong, it might be very good, and always enjoy it. Enjoy the work and enjoy the collaborations. I'm always amused when in movies you see these scientists that are a bit crazy and work by themselves in some obscure lab, because we are maybe a bit crazy, but we work together and we enjoy it immensely. And this idea of the lone scientist is a strong misconception. So enjoy yourself and enjoy your collaborations and do good science. That's my advice.
That's great. And you get lots of opportunities to build new collaborations during NeuroMatch Academy.
Absolutely.
So do take advantage of that. Awesome. Byron, do you want to say a few last words?
Yeah, I think those are great words of advice, Sara. So maybe I'll just briefly emphasize something that I mentioned earlier, which is that it's really easy to take some off-the-shelf dimensionality reduction method and apply it to your data. And that can be very dangerous because the outputs of a dimensionality reduction method depend critically on what that method is actually doing, what its assumptions are, and what it considers to be signal and what it considers to be noise. And so before applying any dimensionality reduction method, it's important to really take the time to understand the method, try it out on some toy data examples where you know the ground truth answer, right? And maybe create data sets that have like similar levels of noise, similar number of time points, similar numbers of neurons as you have in the real data, and check to see whether the method can recover what you expect it to recover. And then only then start to apply it to some real data and doing science with it.
Great, thank you so much. Carsen.
Yeah, that was great advice, Byron. I think it can be useful to use simulated data, particularly if you're developing a new technique for dimensionality reduction. I would add something that we didn't talk about today, which there are dimensionality reduction techniques which take into account the fact that you're trying to predict another variable, such as reduced rank regression. And that's something Byron's worked on as well. And so there are these techniques where you can look at kind of the shared dimensionality of data to an external correlate or neural data, or the other way around as well. Yeah. So yeah, there's lots of different techniques that you can use that I hope you enjoy using, and I hope you have fun using these techniques.
Awesome. Thank you so much, Carsen. And thank you again for all of you to joining us here. I had a lot of fun in the discussion. I hope that that also spilled over, that impression spilled over to you, of course, but also to our students and TAs and other listeners, potentially. So with that, thank you again for being here. Thank you for sharing your time. And I hope you'll see you soon.
Thank you. Thank you very much. Thanks, everybody. Bye-bye.
Yeah, thank you for joining.